Today's lecture is about blood. What is blood? Blood is a very specialized type of connective tissue which is its matrix is formed of fluid. As we know, connective tissue is mesodermal in origin and formed of few cells scattered all over abundant amount of matrix. So as the blood is a connective tissue type, it is formed of abundant matrix, which is a plasma representing 55%, and blood cells scattered inside the matrix. What are the blood cells? Blood cells include red blood corpuscles, leukocytes or white blood cells, and blood platelets. The only true cells are the leukocytes or the white blood cells. The other two elements are not true cells as they do not contain nuclei. How to examine a blood? Examination of the blood is by preparation what is so-called a blood film. We obtain a drop of blood and put it on a slide and spread the blood film by another slide. Let it dry and then stain it with a special stain for the blood called Leishman stain. Leishman stain is a compound stain containing basophilic and acidophilic stains. We are going to talk about the blood elements. First is the red blood corpuscles. Here in your book, page 27, there is a table. I will explain here not in a table form, but you have to study the table. The first column of the table, the RBCs or the red blood cells. They are the most numerous elements. The number of RBCs ranges from 4.5 to 5.5 millions per cubic millimeter. RBCs are only one type. There are no different types of red blood cells. There is one type. They are not true cells, as we mentioned before. They contain hemoglobin only. They do not contain organelles nor nuclei. It is formed of cell membrane enclosing inside the hemoglobin protein of the blood. What is the shape of the RBCs? They appear biconcave. They are biconcave discs. Biconcave discs, thin from in the center and wide from the periphery. In stagnant circulation and in blood films, sometimes they develop rouleau formation. Rouleau formation means they stick to each other over each other, forming a characteristic shape as we know, as we see in the photo. Fresh color is not included in your exam. The size of RBCs, as it is biconcave disc, it has several dimensions. One of them is the diameter. The size of the RBCs ranges from 6 to 9 micrometer with an average of 7.5. Thickness and periphery are just for demonstration. As it is not true cells, they cannot resist changes in the osmotic pressure, and so if they are put in a hypotonic solution, they will rupture easily and they undergo hemolysis, what's called hemolysis. So they cannot resist changes in the osmotic pressure as they are not true cells. They develop from the bone marrow, and the main functions are gas exchange, and they perform their function inside the blood only. They develop in the bloodstream, and they get released to the blood. They develop in the bone marrow, and they are released then to the bloodstream. Their lifespan is 4 months. The other column of the table in page 27 is the white blood cells or the leukocytes. The total number of white blood cells ranges from 4,000 to 11,000 cells per cubic millimeter. Differential count of the white blood cells means the total, the percentage of each type relative to the total number of leukocytes. The white blood cells are not one type. There are 5 types of white blood cells, which are, they were classified according to the presence of the specific granules, the specific stainable granules. Stainable means have a staining affinity. They are classified into 3 types: the granulocytes, basophils, eosinophils, and neutrophils. And the granulocytes, which do not have specific granules, are 2 types of cells, which are lymphocytes and monocytes. They are 2 cells, so they contain nuclei, organelles, and do not contain hemoglobin, of course. The shape of the white blood cells, as they are true cells, they can change their form. They have amoeboid movement. They are rounded, and the surface is irregular. There is no development of rouleau because they are true cells and they can move. Color, they are not included in the exam. Color, they are colorless. The size of white blood cells, as they are 5 types, each type have a diameter variable from other types. So the diameters vary from 8 to 18 micrometer. They are true cells, so they can resist osmotic changes, osmotic pressure changes. They develop in the bone marrow and the lymphatic tissue. Their lifespan varies from days to years according to the type of the cell, and they are present both in the connective tissue and the bloodstream and lymphatic organs and the lymph fluid. Abnormal number and adaptation of the RBCs to perform their function are not included in your exams. What are the different types of leukocytes? First, the granular type. Here we can see the table, and the tables are included in the exam, and I will explain each type separately. The first one is the neutrophil, the first and the most common type, representing 60 to 75% of the circulating white blood cells. The diameter is 10 to 12 microns. It is characterized by the shape of the nucleus. It is one nucleus, but it is segmented or lobulated, formed of segments from 2 to 5 or 3 to 5 lobes, and each lobe is connected to the neighboring lobe by a thin chromatin thread. So it is segmented, connected by thin chromatin thread. From here, it can develop another name. It is polymorph nuclear leukocytes. Polymorph means nucleus with different shapes. The cytoplasm contains 2 types of granules, the primary granules or the lysosomes, which contain hydrolytic enzymes, and the specific granules or the secondary granules. The staining affinity is pale pink. They contain several enzymes specific for neutrophils, including phagocytin, which is bactericidal, lactoferrin, bacteriostatic, and collagenase. The main function of neutrophil is phagocytosis. It performs its function outside the bloodstream. It leaves the blood capillary to reach the connective tissue. Then it phagocytoses the bacteria and the invading organelles. After phagocytosis, as we see here, the enzymes inside the granules fuse with the phagocytic vesicle and to destroy the invading organism. Some of the neutrophil cannot withstand the battle and they become dead. Dead neutrophils containing dead bacteria and damaged tissue forms pus. What's pus? Pus is a viscid white fluid present inside the infected wounds. Pus, what is the importance of the pus? Pus produces pyrogenic factors or pyrogens which elevate the body temperature to inhibit the bacterial growth. Abnormal counts are not included in your exams. The other second type of white blood cells is neutrophil. Neutrophil, the least common type. It represents 0 to 1% of the total circulating white blood cells. The diameter is similar to the neutrophils. It is from 10 to 12 microns. The nucleus is characterized by being S-shaped, irregular. However, we cannot see the nucleus because of the coarse granules, coarse secondary granules, which are masking the nucleus. They are large, basophilic, obscuring the nucleus. Also, we have the primary or non-specific granules, which are lysosomes. The granules of the basophils contains histamine, heparin, and eosinophil chemotactic factor. What's the function of the basophil? Basophils secrete heparin, which is anticoagulant, prevent coagulation of blood in the slow circulation. Histamine, which initiate allergic reaction by increasing the capillary permeability, and secrete eosinophil chemotactic factor. Eosinophil chemotactic factor attracts eosinophil to terminate the allergic reaction. Basophilia is not included in your exams. Eosinophils, the third type of granular leukocytes, the diameter is from 2 to 4% of the circulating white blood cells. The diameter is slightly larger than basophil and neutrophil. Its diameter is from 12 to 14 microns. The nucleus is formed of two lobes connected with thick chromatin thread, peripheral thick chromatin thread, forming horseshoe-shaped nucleus. The cytoplasm contains also primary granules which contain hydrolytic enzymes plus the specific granules. The granules here are large, acidophilic, appearing shiny red. They contain histaminase and heparinase. What's the function of eosinophil? They are concerned with inactivation of the larvae, terminate the inactivation and kill the parasitic larvae, and it has a role in allergy. What is the role of allergy? They are attracted to the site of allergy by eosinophil chemotactic factor secreted from basophil and terminate the allergic reaction by histaminase, which destroy histamine, and finally they engulf the antigen antibody complexes. Here we come to the second type of leukocyte, which is non-granular leukocytes or granulocytes. There are two types of cells. They are lymphocytes and monocytes. Lymphocytes, they are classified according to function into B, T, and TB, and natural killer cells, which is not our focus. We are focusing about the histological types. The histological types are large and small, large lymphocyte and small lymphocytes. The total percent of the lymphocytes is about 20 to 30 of the total number of circulating white blood cells. They also exist in the lymphatic tissue and connective tissue. The small lymphocytes represent 15 to 20% of the total leukocytic count. Their diameter is from 6 to 8 microns. The nucleus occupies most of the cell, rounded and darkly stained. The cytoplasm is scanty, forming a very narrow rim around the nucleus, which is pale basophilic. Here this slide is included in your exam. The small lymphocyte is formed of very small cell comparable to the size of the RBCs similar to the size of the RBC. Most of the cytoplasm is occupied by the nucleus, which is darkly basophilic. The cytoplasm is lightly basophilic, forming a very thin rim around the nucleus. The nucleus is rounded. The larger cell, when it becomes activated, it becomes large. Large, how large? The nucleus, the diameter becomes from 12 to 15 micron. Larger, much larger than the red blood cell. Their percentage is from 5 to 10 in the circulating blood. The nucleus here becomes indented, rounded with indentation, large, pale, with prominent nucleolus. The cytoplasm is abundant and deeply basophilic. The classification of small lymphocytes are not included in your exam. B lymphocytes, T lymphocytes, and so on are not included in your exam except the following slide. This slide classifies the small lymphocytes into B lymphocytes, representing 25% of the circulating small lymphocytes. They develop from bursa of Fabricius. You have to know the site of development. B for bursa of Fabricius. In humans, developed from the bone marrow. It is responsible for humoral immunity. You have to know just B lymphocytes, representing quarter of the circulating small lymphocytes, will site of development, bone marrow in human, and bursa of Fabricius in birds, and its function is humoral immunity. We don't have to know the different types of B lymphocytes or other functions, just humoral immunity. The T lymphocytes, in contrary, represent 75% of the circulating lymphocytes. They mature in the thymus gland, and they are responsible for cell-mediated immunity. So we have to know only their percentage, site of development, and the function, either humoral or cell-mediated immunity only. We don't have to know the abnormal count of lymphocytes. The last cell of white blood cells is the monocyte. Monocyte represent from 3 to 8%, and their diameter ranges from 14 to 18 microns, so it is the largest circulating lymphocyte. By light microscope, the nucleus appears large, eccentric, and kidney-shaped. The cytoplasm, non-granular, non-clear. Non-granular does not contain specific granules. Non-clear because of the presence of the many lysosomes. They give a frosted glass appearance. By EM, by electron microscope, they have pseudopodia, well-developed Golgi apparatus, and lysosomes. Pseudopodia, well-developed Golgi apparatus, and few mitochondria and lysosomes. Function, we have to know just it is a phagocytic cell, highly phagocytic cell. We don't have to know the other points here. Large phagocytic cell, they perform their function after leaving the bloodstream. When they leave the bloodstream into the connective tissue, they transform its name from monocyte into macrophage. They are large because their diameter is from 14 to 18. The small phagocytic cell of the blood is the neutrophil, which is the diameter from 10 to 12. They remain in the bloodstream for 3 days and leave it to reach the connective tissue and stay for 3 months. Here, all we have to know about the blood. Other points not included in the lecture is not included in your exam, and best wishes. Thank you very much.